Today, I'd like to do three things. First, remind you that quiz 2 is due on Saturday. And you have any time between now and Saturday to do it. But once you log in, you'll have 15 minutes. And you'll have 2 attempts. Then we'll discuss quiz 2 on next Monday. I'd like to go over 2 of the worksheets, one of which was just due at noon today. First, and then go on to the topic of the day, which is classification of organisms. You see the worksheet, Methods of Scientific Inquiry?
Yeah.
Okay. So the first question here was, which of these items can be answered through science? And most of you got that all right. The 2 that can't are, how can my team work better together? And is it hot or cold today? All the rest of them can be assessed through scientific thinking. And the second one, I think everyone got right, about graphs. And then which are theories and which are hypotheses. The first 2 are hypotheses. If a hormone is applied to leaves of a plant, the plant will grow faster. That's not well established. Plants grow best in the absence of light, certainly not well established. The next 3 all are. All living things are composed of cells. Some diseases are caused by microorganisms. And matter is composed of atoms. All of those are well established, so they are indeed theories. And the last 3 are all hypotheses. About aphids, moisture and germination of seeds, and apples in the refrigerator. None of them is established well enough to be classified as a theory. And remember that there's no clear line of demarcation between a theory and a hypothesis. A hypothesis which has little or no support is clearly a hypothesis, but then as it gets more and more support through experimentation or observation, at some point it becomes well established, and then it can be called a theory. So there's no clear distinction between the 2. Then we have 2 little stories about Bob and his garden. So what's wrong with Bob's experiment? Well, the growth conditions should have been a constant. Instead, he put one set in a sunny window and another set on a shelf. So they weren't comparable. He didn't control the volume of water that he used to water the plants. So the only variable should have been the fertilizer, and that was not true. And I think most of you said something to that effect. Then Jenny and her snacks. So most of you pointed out that Jenny had no control group not eating snacks. So that is a big deficiency. Secondly, no statistical analysis. So how can we know what's different from what? Well, clearly, 78% is no different from 78%, but is 82 different from 84? Is 64 different from 80? We can't tell just by looking at those numbers. A statistical analysis would let us know within the limits of some significance value which numbers are different from what other numbers. It also would have helped to have to plot the data, but a huge uncontrolled variable here is the lives of the students. So she's just asking, did they eat snacks, and what do they do in the test after they ate snacks? But she didn't control for what did the student eat that day besides snacks. How much sleep did the student get? How much exercise? And on and on. So the lives of the students probably were highly variable, which throws a huge uncontrolled variable into the experiment. Any questions about this worksheet or comments? Anything you don't agree with?
Nope, all pretty straightforward.
Okay. Good to hear that. Then we'll go on to the next worksheet. Which is cell structure and function. You should be able to see that worksheet. Tell me if you can't. I won't go over these definitions of the first four terms. They're all straightforward right out of the book. The next one is a little more difficult. So you were asked to, first of all, identify the three domains of life, and then say what is unique about each according to two different characteristics. So this wasn't easy. And what I got, you can see here in red, comparing according to my characteristic one, bacteria are single-celled and live in temperate environments, whereas archaea are extremophiles and eukarya are multicellular. So by those three criteria, I thought that I could identify a unique characteristic. And for the second one, I focused on spores. So first of all, archaea do not produce spores. Bacteria do, but they have no nuclei, whereas eukarya produce spores that do have nuclei. So I gave credit for whatever you put down, as long as it wasn't wrong, and as long as you indicated what was unique about your characteristic. Anybody have any questions about that? By the way, when I go over these, I edit them in a sense, in that if I have a comment, if I'm going to take a point off, I indicate that on the TypeScript. So if you go to your TypeScript after I've graded it, you should be able to see any comments that I've made. Describe the three main types of prokaryotic cells. You all did that fine, rods, spheres, and spirals. And then draw a picture of a bacterial cell, label it, and indicate the functions of the various components. Well, I did a very crude drawing here, but what the things that I labeled were cell wall, plasma membrane, the nucleoid, cytoplasm, flagella, and ribosome. And for the functions, I indicated the wall is for protection, the plasma membrane for differential permeability with the outside world. Nucleide, of course, is the genome. Cytoplasm is where metabolism occurs. Flagella are for locomotion, and the ribosomes for protein synthesis. Then the next one, again, you had to think a little bit about this. What's the difference between prokaryotes and eukaryotes? Well, prokaryotes have a nucleoid, eukaryotes a nucleus. Prokaryotes have no membrane-bound organelles, and eukaryotes have membrane-bound organelles. And the reason membrane-bound is an important consideration here is that bacteria or prokaryotes do have an organelle, the ribosome, but it's not membrane-bound. Whereas in eukaryotes, the ribosomes are bound in membranes. And we'll talk a lot more about that when we get to nucleic acids. Why do we not find single cells that are so large that we can see them without a microscope? And the important words here are single cells. So we're not talking about why are the cells of a multicellular organism small, but why are cells of single-celled organisms small? And most of you said something about the surface area to volume ratio, which is correct. As that ratio gets, as the cells get bigger, the ratio gets smaller, and that makes it more difficult for them to transport and communicate with the outside world. And the same phenomenon, the area to volume ratio, limits metabolism. As the cell gets bigger, the metabolic rate gets lower and limits the ability of the organism to reproduce or to adapt if the environment changes. And again, on this, I gave you credit for thinking about it and only took a point off if you actually said something wrong. What is a virus? It's a small infectious agent that reproduces only in living cells. And what is it made of? Well, nucleic acid for sure. And most of you said it could be either DNA or RNA. And also they always have proteins and perhaps other molecules like lipids in their envelope. Anything, any comments, questions about this worksheet? Do you find these worksheets helpful?
Yeah, I find them helpful.
Okay, you'll be able to answer that question better after you have a quiz or two. But please, as we go along, give me some feedback about the worksheets, what's useful about them and what isn't. Classification of organisms. That's the topic for today. All right, how many species are we talking about? New species are being discovered all the time. The total number of species on Earth is estimated to be 8.7 million. Notice the standard error here is 1.3 million. So there's really a lot of uncertainty about the total number of organisms on the Earth. So of this expected total, only 14% have been described and cataloged so far, which means that 86% are only predicted. They are not actually known. I just gave you some examples here of species that have been discovered recently, like this hummingbird on the right, a sea slug, the lower left, this psychedelic frogfish, really a weird looking thing, described in 2009. And on the right, this giant salamander is not something new, but this image of a salamander eating a snake is new. And I thought that was an interesting image. So species are becoming extinct faster than they're being discovered. So a lot of species never will be discovered because they're unknown, they go extinct, and we never knew they existed, except perhaps by the fossil record or their DNA imprint. So I have here listed on the left some well-known examples, most of them are well-known, of extinct species going back as far as 1690. The dodo bird was extinct because pigs and cats drove it out of existence. And if you look 1690 and look on this graph, it's way back here when extinction rates were very low. The sea cow, extinct because of its fur and oil. The Labrador duck couldn't compete with humans. Humans ate its food. Passenger pigeon is a very well-known case. The skies would be dark with these flocks of passenger pigeons. No one ever suspected that they wouldn't survive, but they were hunted to the point that finally they all died. They were all consumed. And you can read the rest of these. But look at the graph and notice that it goes back as far as the 15 to 1600s, where the extinction rate was at the base level. And so what is the base level? The baseline extinction rate is one species per million per year. So that's the dotted line here. So the base extinction rate is going up. If you look at, but going up very slowly. If you look at individual groups of species like mammals, the blue line, the extinction rate is rising at a very fast rate. And in fact, they all are, starting in at about the time of the Industrial Revolution. So this is when we started to pollute the planet profusely and started in many undeveloped areas to encroach into the habitat of a lot of different species. So essentially, if you look at all vertebrates, the green line, all of them are going up dramatically starting in the last 2 centuries. So what can we do about preventing extinction? Well, we have to do something about the rate at which the climate is changing and the rate at which humans destroy the environment, the habitat for various species. Another idea, which actually hasn't been done, and we may or may not ever have the technology to do it, is to reverse extinctions by genetic engineering using ancient DNA. So take DNA from a species that is extinct and try to regenerate the organism from the sequence of its own DNA. Just for example, you may remember some time ago, a sheep was cloned from a single cell. So the technology might come along where we can do that. I know there are people trying to regenerate woolly mammoths.
I have a question.
Yeah.
How would that process of regeneration happen, especially with the woolly mammoth and other animals?
Yeah, you'd have to have the complete genome sequence and then translate it. So put it in a cell and try to get that cell to divide. Now, we know you can do that with bacteria. So for example, you can take the genome of a bacterium, E. coli, stitch it all together so that it's a complete sequence, put it into a cell that has no genome. In other words, remove the nucleoid so it has no genome, and get it to grow. So there are components of the technology that's needed. But so the question, for example, with a woolly mammoth, the first technological problem, it would be a huge genome. Okay, and so what kind of a cell are you going to put that in? Because there are no woolly mammoth cells around. So you'd have to put it into some kind of a surrogate cell. And then how are you going to incubate it? Are you going to put it in some animal's uterus? There are just so many things that would have to be worked out like that. So I don't think we're going to see any woolly mammoths soon. But it's fun to think about. Right, so we don't know most of the organisms on the planet, but we do know a lot of them. And so how do we keep track of them? Well, we classify, which that means we divide them into groups. And the groups, by some criterion, show relatedness. And then we give each one a name. And that name has to be unique, and it has to be unambiguous. And so we're going to talk more about that. Humans have been trying to classify the world of biology for a long time. Just give you an example from Aristotle, who lived actually BC, and he based his classification scheme on what he could see. So it was easy for him to notice that there were animals and plants. So that was his first level of classification. And the plants were fairly easy because he classified them by size. The big ones he called trees, the little ones he called herbs, and in between he called them shrubs. Well, animals posed a much bigger problem for Aristotle, but he started out by trying to determine the ones that had blood and those that didn't. And you can see some of the ones that obviously have blood, he has classified together. And then most of these that he says had no blood really do. So this wasn't really a good classification scheme. But it was a start, a way to systematically think about collecting organisms into groups. Now, we owe much of our classification system that we use today to this guy, Linnaeus. His classification criteria were function and form, morphology, the shapes of organisms. But the most important thing he did was propose a binomial method for naming so that we can unambiguously identify any organism that's ever been described. So binomial means that it has two names, and those two names are called the genus and the species. So this is the formal scientific name. And this shows you how it works. On the left, you go from kingdom on down to species. And so this is a hierarchical grouping of what are called taxa. Each one of these is a taxon. And they range from a kingdom, which is the largest and most inclusive group, to the species at the top, which is the smallest, most exclusive group. And this consists of organisms that are only organisms that are similar enough to each other that they can produce offspring together. So if we look at the example of the binomial for humans, Homo sapiens, we start out with asking, what kingdom do humans belong to? Well, they're animals. So these are organisms able to move on their own. Then the next level, humans are animals with a backbone, so they're chordates. The next level, they're mammals, which means chordates that have fur or hair and milk glands. They are primates, which have collarbones and grasping fingers. They're hominids, that's the family level. They have flat faces and three-dimensional vision. Now we get to the real name, genus and species. They're called Homo, the genus, because they have an upright posture and large brains. And the species is sapiens, which means a member of the genus Homo that has a high forehead and thin skull bones. So this is the classification of humans. Every other known organism that's ever been described follows the same pattern, the same set of taxa. So let's look at a couple. What's a binomial for humans? Homo sapiens. And this is the daughter of the former department chair. It's a binomial for your cat, Felis catus. It's a binomial for Shamu, Orcinus orca. These are all genus and species names. Now I put a circle around this and said, important, remember this, because I'm sure I will ask you about it sometime. It has to do with how you write the binomial. Genus is capitalized, the species is in lowercase. So if you take the example of the bacterium, Escherichia coli, you write that out. The first letter of Escherichia is capitalized, big E. The first letter of coli is not capitalized, it's lowercase. And they're distinguished by either being italicized or underlined. So if you italicize, you will have a space between the words. If you underline, there's a space between the words, which is not underlined. So the underline has a gap if you underline. And once a binomial is used in a paper, it starts out, the first time it's used, it's elaborated in full, but after that it's abbreviated. So instead of writing out Escherichia coli every time, after the first time, you'd simply say E. coli. So here's an example. The bacterium Streptococcus pyogenes is known to cause strep throat. The antibiotic amoxicillin can be used to kill most strains of S. pyogenes, not Streptococcus but S, Pyogenes. And you'll notice that it's italicized in this case. Now, humans are natural classifiers. And if you think about it, we classify things all the time. As long as we're awake, every time we look around, we're classifying things. So you're sitting there in your room or office. Look around you. What can you classify?
I have a hat hanging over here.
You have what?
A hat hanging on a hook.
Okay. So you classify things that you put on your head as hats. What about furniture? What about you're using a computer or a cell phone? Could that be classified as what?
A device?
Right, a digital device. Look out the window. What do you see?
Houses,
Okay, structures known as houses. What else?
trees,
Trees. Right. So my point in asking you this is to just make you aware of the fact that every time you look at a scene, at an array of anything, you classify it automatically in your mind. So it's only natural that we try to make sense out of our classification. Aristotle's system obviously didn't work. Linnaeus's was much better, but he didn't have the technology we have today. So today we classify primarily on the basis of ribosomal RNA sequences. And I'll tell you a little more about that later. But using ribosomal RNA, we've come up with 3 domains of life: bacteria, archaea, and eukarya. And the eukarya are the ones with nuclei, and they're divided into plants, fungi, animals, and protists. Protists I'll talk about later, but it's in general, anything that doesn't fit any other category is classified as a protist. So bacteria are prokaryotes, as are archaea, whereas the eukarya all have nuclei, eukaryotic cells. So let's look at how we go about this. We start out with domains. We have 3 big boxes, one for each domain. We can take a particular domain, like eukarya, and divide that into 4 boxes. And we're going to continue to subdivide. Take the kingdom Animalia, so we've just taken one of these boxes, animals, and we can go through kingdom, phylum, class, order, family, genus, and species. Here are all the phyla in that group. And this is the complete sequence, which you should remember, starting with the domain, kingdom, phylum, class, order, family, genus, and species. Usually people develop some kind of a mnemonic so that they can remember the order of these taxa. So let's go through a classification of 3 species to see how it works. So we have 3 species. We classify them according to domain. They're all eukaryotes. The kingdom for all of them is animal. The phylum is arthropod. When we get down to class, they segregate. So one species is a crustacean, two are insects. Order, one is Decapita, the other two are Diptera. We get to family, one is Charadia, Drosophilidae, and Matacera. So they're all, each is different at the level of family. And then when we get to genus, they're all different, Homerus, Drosophila, and Aedes, and they're different by species, Americanus, Melanogaster, and Egypti. So what are these? Well, Homerus is a crab. Drosophila melanogaster is a fruit fly. And Aedes aegypti is a mosquito. Now, I'm going to tell you a joke. You may not think it's funny. You don't have to laugh, but it amuses me. It's about the fruit fly. The fruit fly is a genetic model, has been for over 100 years, used in labs around the world. So it's a very popular organism to study in labs. As you go around from university to university in various different labs, if you go into a fruit fly lab, you'll almost always see a particular sign hanging on the wall. And then in one case, they had an additional sign, which you see on the lower right, which goes even further. I think that's funny because it's weird. It may or may not have made you smile, but now you know about fruit fly labs. What criteria are used to classify organisms? Well, here you see pictures of wings. So you could say wings could be used to classify organisms, but it doesn't work. It breaks down rather quickly, as we'll see in a minute. Classification is now based on what's called phylogeny, which is the evolutionary relationships between organisms. And how do we track evolution? We track it by ribosomal RNA sequences. And I'll tell you how that's done in a little bit. But first, let's think about wings. And in doing so, I'm going to introduce you to two terms, which you'll need to remember. They're analogous and homologous structures. So let's first look at the bat wing, the bird wing, and the insect wing. They're all used for flying, but where did they come from evolutionarily? Well, it turns out if you look at the bat wing and the bird wing, all the bones are the same. So you can, and they're color coded here. So the red bone in one is the red bone in another, yellow bones, purple bones. And so since in two completely different organisms, the structures are the same, they're called homologous. Now, if we look and compare either of those with an insect, none of the supports in an insect wing have anything to do with the supports in bird wings and bat wings. So they are called analogous because they have the same function, but completely different origins. And in the upper left, you can see this laid out in more detail, how bat wings and bird wings are the same structure, but on two different animals. And how does this happen? Well, if you look at this scheme here, the phylogenetic tree of homologous structures with wings, it starts out there's a common ancestor long ago that had 4 limbs. And that 4-limbed creature evolved, split off a branch that had birds and crocodiles. Birds and crocodiles are closely related, relatively speaking, closely related. One has wings and the other doesn't. Now, the other branch of this tree has a mouse and a bat. Both, they are closely related. Bats have wings, mice do not. So that is how bat wings and bird wings turn out to have homologous structures. So the definitions are here. Analogous is similar function, but not evolutionarily similar. Homologous is that 2 structures have a common evolutionary history. So bird wings and bat wings go back to a common ancestor. So is that clear? Any questions about this? You should remember these terms, homologous and analogous, and what the difference is. The contemporary criteria for classification are, to some extent, still morphology. We still ask, what's the shape or the form of organisms? But more importantly, is ribosomal RNA sequence. Why is ribosomal RNA so useful? Well, it's unusual in that it's found in all organisms, from prokaryotes to archaea to eukaryotes. They all have ribosomal RNA. It's because they all do protein synthesis, so they need ribosomes. Some sequences, regions of the sequence are highly conserved, and some are highly variable in the same molecule. And you can see that in the lower left here. This is the structure of a ribosomal RNA molecule. And you can see that this is the amino acid sequence. You can see it's color coded. So the red ones are highly conserved. The blue ones are not conserved at all. And you can see that both are distributed throughout. So if you do an analysis like this with ribosomal RNA genes at the nucleotide level, you see what's on the lower right, where the top 3 sequences are from mammals, 1 from human, 1 from mouse, and 1 from rat. And the lower 3 are from bacteria, 3 different bacteria. The yellow nucleotides are similar in all, which means that they are essential, probably essential for all life, since they're so highly conserved between mammals and bacteria. The differences are in red and green. So the mammals all have a certain set of green sequences, none of which show up in the bacteria, and instead they have red sequences. So using these sequence similarities and differences, it's possible to classify with great precision all organisms, from prokaryotes to eukaryotes. And by doing that, people build what are called phylogenetic trees. And this is a phylogenetic tree of life, of all of life. Starting with, I've circled down here, the hypothetical progenitor cell. So as far as we can tell, life has evolved on this planet just once, and it started with a cell somewhere back in the primitive ocean. So we have bacteria on the left. archaea in the center, and eukarya on the right. I won't say much about these. The archaea, as you know, are extremophiles. They live in extreme environments. So halophiles, for example, live in highly salty environments. Methanophiles produce methane. Thermophiles like it hot. And you notice that on the right, the organisms that we are most familiar with are a tiny fraction of the tree of life, the animals, plants, and fungi. I've made 3 more circles: the plants, the green filamentous bacteria, and the cyanobacteria, because all life on Earth depends on them, and that's because they photosynthesize. They convert the sun's energy into chemical energy. And we'll talk mostly in this course about plants, but keep in mind that not only plants, but certain bacteria can also make food. And everybody else eats it. So let's talk about bacteria. They're prokaryotes, variety of shapes, and we've talked about this before. There are 3 basic shapes: the spirals, the rods, and the spheres. They're found everywhere. Some are pathogenic. The domain archaea, these are a little harder to get your teeth into. They are prokaryotes. They're always single-celled. And they almost always live in extreme environments, like in a thermal vent. I once worked at a biotech company that isolated bacteria from extreme environments. We actually had one that would not grow at a temperature lower than boiling water. So it was really hard to keep this thing going, because if you lowered the temperature to something reasonable, it just stopped. So the archaea are everywhere. Most of them are not culturable. I mean, this poor thing that grew in boiling water was culturable under extreme conditions. So how do we know that the others exist? Well, a process known as metagenomics, which means that you take an environmental sample. So for example, a handful of soil, and you extract all the nucleic acids from it, purify them, and sequence them. And from the sequences that you get out of a crude sample like that, you can allow computers, using proper algorithms, to stitch them together and simulate the genomes of organisms that produce those sequences. So you know they exist from the sequences that they left behind. The archaea can be found all over us. None of them is known to be pathogenic, however, so we don't have to worry about them. They can be infected by viruses. So a lot of thermophiles have been isolated from hot springs or thermal vents, or those little smokers that you see pictures of from the bottom of the ocean. Some live in very acid environments. Some live in very salty environments. And they have various different shapes. And so let's switch now. We've done bacteria and archaea to eukarya. These are primarily plant and animal cells, but also include protists and fungi. Here you see pictures, animal cells and plant cells. The protists is a weird group. So the protists are everything that doesn't fit easily into any other category. So for example, an amoeba is a protist. Notice it has a prominent nucleus, so it is a eukaryote. And again, just more examples to give you some feel for what this broad category involves. Here are some of the types of organisms that are protists. Paramecium, you may have studied this in your high school biology lab. Here's an alga. Here's a kelp forest off the coast of San Diego. Here's an organism that looks like a fungus, but it isn't. It's really a protist. And now I'd like to tell you a couple of little stories about fungi. Highly diverse. They have their stomachs located outside of their bodies. So to obtain food, they secrete enzymes out into the environment. The enzymes that they secrete break down whatever organic molecules they find into small molecules so that the fungus can then absorb those small molecules and use them as nutrients. So they're very unusual in that regard. And now I'd like to tell you, remember the first class period, I gave you that quote from Isaac Asimov, that most big discoveries are not recognized as aha, but rather, wonder why that happened. Well, the story of penicillin is one of these moments. And it goes back to a man named Alexander Fleming, who lived from 1881 to 1955. He discovered penicillin in 1928 and won the Nobel Prize for it in 1945. So how did he do it? Well, Fleming, here you see a picture of him in his lab with petri dishes and test tubes, and here's a colony of penicillium. He's a bacteriologist, which means that he was interested in bacteria. And he was doing experiments with bacteria by spreading the colonies out on petri dishes containing agar medium, letting them grow and observing them. And he came back one day, and one of the worst things that can happen to a bacteriologist is he found a fungus growing in his petri dish. So this has been happening to bacteriologists for 100 years. And when this happens, you throw out the plate. It spoiled your experiment. But Alexander Fleming said, so you look more carefully at this plate, and he noticed that there's this zone between the fungus and the bacterial cells where the bacteria are not growing. So after thinking about that for a while, he made a hypothesis. And his hypothesis was that the fungus has produced something that's killing the bacteria. And that hypothesis turned out to be correct. And the substance turned out to be penicillin, which is still today one of the main antibiotics, that and derivatives of it, that we use. So this is a photograph. He saved the original plate where he observed the effect of penicillin, took a picture of it. So the picture is here, and he wrote on it, at the bottom, he says, a large penicillium colony at the top, and the staphylococcus colonies showing degeneration. And at the top, he has written the original culture plate on which penicillium was observed. And I have here pictures. Here's penicillium growing on a plate of agar, a pure culture of it. So this would be the same fungus that Fleming saw growing on the edge of his plate. And these are the body, the hyphae, and the spores of this fungus. Fungi interact with humans all the time. They're the great degraders. As I said, they secrete, wherever they are, they're secreting enzymes and digesting things. If it weren't for fungi, the world, the whole planet would fill up with detritus. But fungi degrade. Everything that isn't being used gets degraded, usually by a fungus. You've all seen something like this in your refrigerator, where you have a fruit or a vegetable that's covered with a fungus. This fungus could well be a species of penicillium, or it could be something else, but penicillium is of this type of mold. Now here's an interesting fungus which catches nematodes, little worms. These little worms grow in the soil, or yes, they grow and they move around in the soil. And this fungus catches them in traps. So each trap, you can see 3 traps here. Here's one, here's another, and there's another over here. Each one is a ring. And if the nematode, the little worm, sticks its head in the ring snaps shut, and the cells, are 3 cells in each ring, start to secrete enzymes, and they literally digest the worm and and they absorb it and grow more and make more rings. Here you see another ring has the nematode by the tail. So this is a highly specialized way of getting food. More familiar to us are the ways that fungi help us get food. So cheese, here you see fungus making cheese. We use yeast to bake bread. Yeast is a fungus. And to make beer and wine. And sometimes we just eat the fungus directly. The largest living organism on Earth at one time was thought to be a fungus. So I have Earth 1 here. So this is a fungus called Armillaria gallica. Was found in the Upper Peninsula of Michigan. It occupies 173 acres, which is huge. It's about the size of the Pentagon. Here you see the Pentagon. It's about 2,500 years old and it weighs 440 tons, whereas a blue whale only weighs 220 tons. And its nickname is humongous fungus. So this is all one organism. So here you see part of the organism. It's a network of strands growing underground. And then when it wants to reproduce, it sends up these little mushrooms, which have inside the spores, the reproductive structures. Here's a close-up of the mushrooms. It's what it looks like growing in a petri dish. So this was touted for a long time as being the biggest organism on Earth. It's all one organism. But now we're going to turn to plants, and we'll talk a lot more about plants and photosynthesis later. But there's another claim for being the largest living organism on Earth too. This is a stand of aspens called the Pando aspen clone. It's found in Utah. It occupies 106 acres. It's about 80,000 years old and it weighs 6,500 tons. And it comprised of 47,000 aspen trees. And all of these trees, which you see here in the picture, they're all actually clones of the same tree. So if you add all these trees up and all their root systems up, it comes to 6,500 tons. It's a huge organism, but it's all one organism. Its future is dismal. The reason is that aspen trees are short-lived. They die after about 150 years. So they have to be continually producing more shoots growing up from the roots of the old trees. However, the shoots are a delicacy for deer and cattle. So whenever a new shoot comes up, something almost immediately eats it. And the deer are prevalent in this area because no deer hunting is allowed. And there are no predators for the deer, which would be the wolves and bears, because civilization has encroached. Cattle farmers don't like wolves and bears, so they've been eliminated. Which means that over time, if these old trees as they die are not replaced with new shoots, eventually this clone is going to die out. Okay, that's about plants. And this is the last slide. Just to remind you that the other eukaryote category is animals. Animals come in all different sizes and shapes. I'm going to stop at that point. I'm out of time because I can only keep you 15 minutes less than normal because you have a quiz, which will take 15 minutes of your time. And so I subtract that 15 minutes from a class period. And as I said at the outset, the quiz is available now. I wouldn't recommend you take it until you've had time to study a little bit for it. It's available until Saturday night. So you have from now till Saturday night to take the quiz. Hey, any questions or comments about how we classify organisms? If not, I'm going to